The first of our speakers for the keynote session is Professor Sir Martin Evans. His lecture will be on embryonic stem cells. He is the winner of the Nobel Prize in Medicine in 2007, Director of the School of Biosciences and Professor of Mammalian Genetics, Cardiff School of Biosciences in the UK. Professor Sir Martin.
I thought that my part in the whole business, of course, is the mouse embryonic stem cells. But I thought this morning, particularly because a lot of the biomedical focus is upon the idea of using stem cells in therapy, that I should say a few words about that, and I hope a few words which may give people ideas and also give people perhaps pause to think about some of the over-optimistic hype that's going on in this area. Now, stem cells, everybody is talking about embryonic stem cells and are realizing that there are also plenty of other stem cells in the body. But I think what we need to consider is that this has been going on a long time. And I'd like to first of all highlight that in 2005, the Albert Lasker Award was given to McCulloch and Till. And these are the people who, starting some 45 years ago, showed that there were cells in the bone marrow which were the precursors of all of the blood cells, both the red and the white cells in the blood, and showed by a series of very elegant experiments that these cells would repopulate themselves. In other words, they were continual resource. They were there in very small numbers, but that they and their progeny could give rise to all the blood. And these are the basis of the ability to do things like bone marrow transplants. Now, they have really set the scene, and really the probably, if you talk about a stem cell in the body, it's the hematopoietic stem cell and the way that that is maintained, the way that that develops through a series of proliferative precursors down various pathways, which is really the model, which is by far the best researched and understood, and a model that we should go back to and look at when we're considering the ability of other stem cells in the body, particularly the stem cells which are committed to particular pathways. Now, they, in their publication, which went together with this award in 2005, they said quite clearly, why had they only been acknowledged now? And they pointed out, as I've highlighted here, that their work, and that's knob on the top side there is a mouse spleen, and the lumps in it are the repopulation of the spleen with a small number of individual stem cells, and that is the basis of their transplantation assay. Why has this come up? And it's because now we're interested in various categories of stem cells. And as they put at the bottom, another reason is the exciting possibility that embryonic stem cells might provide a means to regenerate damaged organs. Now, on the bottom left there is a picture of a bone marrow transplant. Top right is a picture of a stem. That is a stem in the plant. In both cases, of course, we have stem cells there. We have in the plant cells which we would recognize as stem cells in the phloem of the conductive things. And here, somewhere buried in there, are stem cells, the hematopoietic stem cells of Till and McCulloch. Now, it's often said that the primal ability of a stem cell is it doesn't divide either to give two cells the same or two cells different from itself, but it can do one of each. And that's a model that I think comes from Drosophila genetics, which may or may not apply in many cases. It's much more likely that in the real world, what we have is a situation more like this. We know that populations of stem cells abide in what we call niches. They are in particular circumstances that maintain them, growth factors and so on, interactions, and then they can move out from that niche and differentiate. And it's quite possible that we are not always talking about commitment at division. Now, one of the things I try to say, particularly to lay audiences, this is, I understand, a very eminent and not a lay audience at all, but is to explain that the structure and function of the body depends upon the function of a set of semi-autonomous units, the cells, which all work from the same instruction set, but have different functions. And they've all developed from the single fertilized cell, which is the fertilized egg. Sorry, single fertilized egg, which is a cell, I should say. And by proliferation. So it's the daughter cells and the subsequent generations which take on new function. And usually this is a tree of decision making which doesn't allow for a change of decision. We know from much work in experimental embryology that effectively there is a stability of the differentiated state. Cells become committed to particular lineages. And by and large, we don't get large changes across these lineages. I'll come back to that later on. At the very earliest stages of development, clearly there must be cells, down at the bottom left of my diagram, which have wide ranges of ability. These have been found in mouse and man, other mammals, and are the embryonic stem cells which we talk about. There are also, along that pathway, other points at which populations of cells may be marking developmental time. These are committed stem cells. And just go back to embryonic stem cells. When I isolated them, we were looking at finding an in vitro developmental system that we could study the processes of cellular development and commitment in tissue culture. That we can from these embryonic stem cells. But almost we and the entire world was diverted by the wonderful opportunity, once we got them working, of being able to use them as a vector towards genetic studies in mice. And of course, they've now been used for looking at experimental modification of probably a third to a quarter of all of the mouse genes already. So the emphasis of use of the mouse embryonic stem cells has very largely been on the experimental mammalian genetics. Now, when Jamie Thompson first isolated the equivalent human embryonic stem cells, he laid out very clearly in his initial paper an extra vision for their use. And that was that they wouldn't be available as he was hoping some of his non-human primate cells might be for an experimental genetics. That hasn't been used, but he was quite clear that the human cells weren't being used there, but perhaps they would be extremely useful in the other side, not only in just the ability to study the processes of cell commitment and differentiation, which can also be done in the mouse, but because they're human cells, the ability to maintain large numbers of them and then to differentiate them into a wide diversity of different cell types might provide the opportunity of using some of those precursor and differentiated cell types for tissue therapies.